Hey friends, welcome you all to Bioskul. This is Gayatri. So in this session, we'll discuss GATE 2020 Life Science paper, and the subject is Biochemistry. So if you are a life science student, then you must watch the video till the end, because we don't know, sometimes we may face similar type of question in any type of examination. So let's get started. The first question: which one of the following hormones initiate a signaling cascade by directly binding to an intracellular receptor? Okay, so in this question it is asking about which hormone it directly binds to the intracellular receptor. So let's see. First one is the insulin. You know, the receptor for insulin is a receptor tyrosine kinase, that is RTK, and receptor tyrosine kinase, it is the cell surface receptor. So this one is not correct answer. Gonadotrophins are like a luteinizing hormone and the follicle stimulating hormone, and the receptor is a gonadotrophin releasing hormone receptor. It is found in the pituitary, that is a GPCR, G protein coupled receptor. So that is also the cell surface receptor. So this option is also wrong. Third option, it is the progesterone. Progesterone hormone are all the steroid hormones. You know, their receptor is present in the cytosol. So when the hormone it is absent, then the receptor is present in binding with a heat shock protein. Okay, so let's see. So see here the steroid hormones, the receptor is known as the steroid hormone receptor. It is present in the cytosol in bind with heat shock protein. So when hormone it binds to the receptor, then heat shock protein it gets released. Then the hormone with the receptor is it's a homodimer. It binds to the hormone response element, that is HRE. Okay, so it is the correct answer. The progesterone hormone or all the steroid hormones, they have the intracellular receptor. Okay, the fourth option, it is epinephrine. Epinephrine, the receptor is GPCR, and GPCR it is a cell surface receptor. So the correct answer is progesterone. Okay, next one: which one of the following bond is not present in ATP? Okay, so first we will see the structure of ATP. So it is the structure of ATP. ATP means adenosine triphosphate. So ribose, it is the sugar molecule. It is the carbon number 1 or the anomeric carbon. It binds with the nitrogen of adenine, okay, by a bond. That bond is called the N-glycosidic bond. N-glycosidic bond, it is formed between the anomeric carbon of ribose with the nitrogen group of adenine, that is the N-glycosidic bond. Similarly, the bond which is present between the sugar molecule and the phosphate, that bond is known as the phosphoester bond. Okay, and in between the phosphate groups, that bond which is present, that is called the phosphoanhydride bond. Okay, so what bonds are present? Phosphoester bond it is present, phosphoanhydride bond is present, N-glycosidic bond is present, but alpha glycosidic bond it is not present. And in question it is asking about which of the following bond is not present in ATP. So the correct answer for this question is alpha glycosidic bond. Alpha glycosidic bond, it is a O glycosidic bond. It is formed between the anomeric carbon of one sugar molecule with the oxygen atom of hydroxyl group of and the sugar molecule. Okay, and this type of bond you will find in the maltose. You know, in the maltose there is a presence of alpha 1,4 glycosidic bond. Okay, so the correct answer is alpha glycosidic bond, which is not present in the ATP. Let's see this one. The reaction involved in the conversion of N-phenylalanine to L-tyrosine is. Okay, so if you know the structure of L-phenylalanine and L-tyrosine, then you can easily solve this question. Both are structurally similar, except in the tyrosine you will find a hydroxyl group. Okay, see here, this one is the phenylalanine and this one is tyrosine. In the tyrosine there is presence of a hydroxyl group. So if there is a hydroxyl group is present in the tyrosine, then which enzyme it will help in the hydroxylation? That is hydroxylation enzyme, phenylalanine hydroxylase. This enzyme it add the hydroxyl group. Okay, so the correct answer will be hydroxylation. By hydroxylation, L-phenylalanine it get converted into L-tyrosine. This question: the human major histocompatibility complex, MHC. Okay, in human MHC, it is known as the human leukocyte antigen, that is called as the HLA, and it is present on chromosome number 6 of human. So, the MHC of human is polygenic. Okay, let's consider if it is a class 1 MHC or class 2 MHC. Polygenic means the presence of many genes. Class 1 MHC has A, B, C genes are present. Similarly, in class 2 MHC, you will find this one is DP. It's not visible, right? So, here DP, DQ and DR. So, many genes are present. So, the MHC, it is polygenic. So, let's eliminate these two options. Then, next one is MHC, it is monomorphic or polymorphic. MHC, it is polymorphic. Okay? So, let's eliminate this one. This one is the correct answer. Polymorphic means different allele are present in different individuals of population. During the organ transplantation, it's highly essential to match the MHC. Otherwise, that will lead to the rejection. Okay, during the organ transplantation, it is highly essential to match the MHC between the donor and the recipient. Otherwise, that will lead to the rejection of organs. Okay, so the MHC molecule, it is polygenic, also it is polymorphic. Hergovin, Khorana and Narenberg elucidated the genetic code by using a cell-free protein synthesizing system. It was found that poly-U and poly-C result in synthesis of poly-L-phenylalanine and poly-L-proline respectively. Based on these observations, which one of the following conclusion is correct? Okay. If there is presence of the poly-U, then this codon poly-U, it will specify phenylalanine, right? Similarly, poly-C, it will specify which codon? This codon poly-C codon, it will specify which amino acid? It will specify amino acid, that is proline. Okay, so the correct answer, it will be option 4. Codon U, triple U, it specify the phenylalanine, and codon triple C, it will specify the L-proline. Okay, if it is the triple G, then it will specify which amino acid? That is glycine, right? It will specify amino acid glycine. Similarly, triple A, it will specify which amino acid? It will specify amino acid that is called the lysine. Okay, so now move to the next question. Okay, binding of an antibody to its antigen does not involve. So, the antibody and antigens, they are bind by the non-covalent interaction, that is a weak interaction, like electrostatic forces, van der Waals forces, hydrogen bonds and hydrophobic interactions. So, not by the covalent bond. Okay, so the correct answer, it will be covalent bond. So, the binding of an antibody and antigen does not involve covalent bond. They are always bound by weak interaction or non-covalent bonds. Okay, let's see the next question. A globular protein of molecular weight 50 kilodalton, okay, exists as a mixture of monomers and dimer in solution. Okay, so this globular protein, it has the molecular weight is 50 kilodalton and it is exists as a monomer or it is exists as a dimer. If it is exists as a dimer, then what will be its molecular weight? Of course, its molecular weight will be 100 kilodalton. 100 kilodalton. So, the most appropriate technique for the separation of these two forms of protein is, okay. So, this question, it is about the chromatography. So, in chromatography, which chromatography it is used to separate the protein molecule based on their molecular weight? That is gel filtration chromatography. Okay. In this chromatography, the protein molecule which has a higher molecular weight, that will elute first. Okay, so the correct answer will be gel filtration chromatography. Okay, in ion exchange chromatography, they are separated based on the charge of the substance. So this one is wrong, and this is not the paper chromatography and thin layer chromatography. So the correct answer is gel filtration chromatography. Okay, let's see this question. Choose the correct order of molecules according to their ability to diffuse across a lipid bilayer. Okay, so in a lipid bilayer, it is a semi-permeable membrane, and through the semi-permeable membrane, first gases they will pass through, then followed by the gas. Which one will be passed through? That will be the smaller on-polar, that will be the small uncharged polar molecules, then followed by the large uncharged polar molecules, then ions. So first it will be the hydrophobic molecule or the gases, so it will be the carbon dioxide. Then the second one will be small uncharged polar molecules. Small uncharged polar molecules are water, urea, so it will be next will be the water. Then next is the large uncharged polar molecule. Large uncharged polar molecules are glucose, then ions. Okay, so ion, it is the Rn, it is a negatively charged, so the correct order will be carbon dioxide, then it will be water, then glucose, then RNA. Okay, so this one is the correct option. Okay, let's say this one. When one glucose unit from glycogen get converted to lactate, here in question, see, one glucose unit from glycogen it converted into lactate in the muscle, so it is about the anaerobic respiration. But see, it asks that glycogen is one unit is converted into glucose. So glycogen, by the presence of enzyme, that is glycogen phosphorylase, if there is a, consider there is a presence of N molecule of glycogen, then by the enzyme glycogen phosphorylase, it will produce one product, that will be the glycogen N-1, glycogen N-1, and another product, it will be the glucose, but here the glucose, it will be released in the form of glucose 1-phosphate. Okay, it is released in the form of glucose 1-phosphate. So here the glucose which is produced, the ATP molecule of the glucose, it will be converted into the pyruvate, then into the lactate, but the glucose which is released from the glycogen, it is in the form of glucose 1-phosphate. So what will form? Glucose 1-phosphate, by presence of enzyme mutase, it will converted into glucose 6-phosphate. Okay, then glucose 6-phosphate to the fructose 6-phosphate, fructose 6-phosphate to fructose 1,6-bisphosphate, then glyceraldehyde of 3-phosphate, glyceraldehyde 3-phosphate, it form the 1,3-bisphosphoglycerate, then 3-phosphoglycerate, then 2-phosphoglycerate, then it will be converted into phosphoenol pyruvate to pyruvate. So in this process, there will be production of 4 ATP molecules, 2 ATP molecule it will be produced in the conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate, and another 2 ATP molecule it will be produced when phosphoenol pyruvate it get converted into pyruvate molecule. 4 ATP molecules are produced, but how many ATP molecules are used here? Here only 1 ATP molecule is used in conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Okay, 4 minus 1 equal to 3. So net number of ATP molecule produced is 3. Okay, if it is the glucose that converted into glucose 6-phosphate, then there will be utilization of another molecule of ATP. But is the glucose unit which is released, that is from the glycogen. So from the glycogen, the glucose unit which is released, that is in the form of glucose 1-phosphate. So when the glucose 1-phosphate is converted into glucose 6-phosphate, then there will be no use of ATP molecules. Okay, so the correct answer it will be 3 ATP molecules will be produced in this process. Okay, this question, considering the 3 pKs of histidine. So histidine, it is a basic amino acid, and its pK are given, pK1 1.8, pK2 9.2, and for Argen it is 6. Its isoelectric point will be, so how can we calculate the isoelectric point? It is a basic amino acid, so we will take the average of 2 basic pK, right? Okay, so average of 2 basic pK is pK2 plus pKr is the 9.2 plus 6.0, 9.2 plus 6.0 divided by 2, so answer it will be 7.6. Thank you so much for watching this video I'm expecting that this video will definitely help you a lot. If you are new to my channel and have not subscribed yet, then please do subscribe for getting such more informative videos in upcoming days.